In Chapter 12, we talked about Mendel's experiments, which led to a theoretical understanding of genetics, but it still didn't explain what the actual genetic material was and how this actually functioned inside of a cell. So in Chapter 13, we're going to dive into a little bit of the early work that started linking genetics with chromosomes, which seems fairly obvious to us, but was not by any means obvious to early scientists. So we'll talk about the chromosomal theory of inheritance and genetic linkage, and then we'll talk a little bit about how inherited disorders are controlled by certain chromosomes. So we're going to talk first about Sutton and his chromosomal theory of inheritance. We'll talk about linkage and how recombination functions with linkage. So remember, Mendel didn't know anything about chromosomes. He could not observe them. He didn't have microscopes that were powerful. And it wasn't really until the late 1800s that we could observe cell divisions and see chromosomes in there. But even then, it wasn't fully clear that they were the genetic material. Mendel did all his work figuring out all his rules just using math and logic. So one of the early studies on chromosomes was done by Bouvier. Bouvier was using the model system of sea urchins. Now, this seems strange to us. Sea urchins are kind of obscure, but sea urchin embryo development was really nice because they're very, very big cells. They're nice and clear, and he could clearly see the chromosomes in there. And what he noticed is that sea urchin embryo development wouldn't occur properly without chromosomes present. It wouldn't occur at all. So he started making the link here between chromosomes and proper development. Again, this is not genetics, right? But this is saying these chromosomes are indeed important. It seems like a baby step, but that's really, really important to understand that, hey, these things are needed for development to occur. Now, we're not going to go into great detail about this, but if you want to learn more about these discoveries, my Micro 250 class, we talk all about this discoveries of chromosomal theory and inheritance and more about the experiments of Bouvier and Sutton, who we're going to talk about right now. So Sutton, independently of Bouvier, showed that chromosomes move in meiosis, and he noted that the gametes that are produced have half the number of the chromosomes that the zygotes that are produced will have. So you have two gametes, they fuse together, and you end up having double the genetic material in the zygote. Again, from our understanding, we know that. I've spent several slides telling you all about diploid and haploid and things like this, but this is very, very early. So Sutton says, I may finally call attention to the probability that the association of paternal and maternal chromosomes in pairs and their subsequent separation during the reducing division, that's meiosis, as indicated above, may constitute the physical basis of the Mendelian law of heredity. That's like a mic drop moment right there, right? Sutton is saying, hey, I'm telling you that I think chromosomes are why Mendel's rules happen. That's pretty big. And that's a big statement. When you make a big statement in science, you need evidence. So what's the evidence? So chromosomes are the genetic material responsible for Mendelian inheritance. That's the claim. Let's find evidence to support this. Well, in meiosis, homologous chromosomes migrate independently of each other. That would make sense given what we observe in genetics. Chromosome sorting from each pair is random. We said that was one of Mendel's rules as well. Each parent synthesizes gametes with only half their chromosomal complement. That's a big one, right? That shows us the Punnett square functioning. Sperm and egg differ in size. That was a clear, sticking point. The egg is so much bigger than the sperm. It clearly must contribute more. But when you examine the number of chromosomes in there, each contribute equally to the genetic material. That's a big one, right? Because we said in the Punnett square, there's an equal chance of either allele from either parent. There's no bias towards any of them. And finally, gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as the parents. That gets at Mendel's hidden units of inheritance. That sounds pretty good, right? Like everything's seeming to line up. But again, you have to remember that you've learned this material already. We've had over 100 years now of understanding how this functions. Not everyone was convinced. There's not any real experimental evidence in there. That's just all correlation. It's strong correlation, but correlation is not causation. So it would take a little bit longer and some more work to show that this is indeed the case. So one of the big sticking points is there are way more traits than, chromosomes. So how does independent segregation happen? How do we explain this? And this is where Thomas Hunt Morgan comes in and his co-workers, his students. He used Drosophila melanogaster, the fruit fly, very important model system for genetics. It has a nice short generation time of 10 days. And he worked at Columbia University in New York. And here is the fly room. Each of these flasks here contains a number of flies in there. And flies are really nice because you can actually soak a cotton ball in a small chemical, and it's called fly nap, and it basically knocks them out. And then you can pick them up, put them in a jar, put them together. They'll breed for you. They'll do all these things. Flies have some nice traits that can be observed as well. And one of the first things that was observed was an X-linked trait. That's really important because the X chromosome could easily be observed under a microscope. It had a nice, distinct shape that was in there. So the X chromosome could be observed, and we found that there was a trait, eye color, that was linked to the X chromosome. So I'm not going to explain the cross here. It's not super important at this level. You'll go through it in 300 level genetics if you take that. But what is important is that there was a trait that we could show moved with the X chromosome. And that was good evidence that a chromosome is controlling a trait. That's experimental evidence right there. But he found some contradictory evidence to Mendel. He found that some traits didn't assort independently. So here we have some different traits. Long legs, gray body, red eyes, short legs, black body, purple eyes. And he found that these traits tended to move together. They didn't completely independently assort when everything was said and done in a single cross. So what do we call this for traits that are on the same chromosome? We call that linkage. Linkage is what they discovered. Some traits on the same chromosome move together sometimes, and they stay together, but not always. So here's two forms of each trait type. Here's the wild type, and here's the mutant type. Things like long wings versus dumpy wings, or black body versus gray body, long wings versus vestigial wings. These traits, they could create a map of this. And to figure out how these related, we have to talk about another process called recombination. So these traits don't always move together. Even though they're on the same chromosome, sometimes you might get long wings with purple eyes. How does that occur? Well, through observations of chromosomes, Franz Alphonse Janssens, he noticed that at certain times, chromosomes kind of formed this X shape that he called the chiasmata. And the chromosomes seem to exchange bits. Okay, interesting. How does this match up with Morgan's work? Well, this can help us explain how we can get what we call non-parental types. Okay, this is the key takeaway here from this chapter. Linkage is traits together, right? So in any cross, we have what we call a parental type. This is the combination of alleles found in the parents. So in this cross, we have two parents, right? We have a gray body with long wings and a black body with short wings. That's the parental type. The recombinant type would be the alternate mix of this, right? Like gray body with short wings, and black body with long wings. That's a non-parental type. These are going to be, in the case of linked genes, the result of homologous recombination. Now, in my example here, I'm showing you that it could be independent assortment. They're on separate chromosomes. But if not, if they are indeed on the same chromosome, the only way to get these new combinations is through recombination. All right, so let's examine what's actually happening in the process of recombination, and then we'll examine how that impacts what we observe in a cross. Okay, so in any cross, we're going to have parents that go into it. Remember our monohybrid cross, we have our two true breeding lines. Now, I've drawn their genotype a little bit differently here because I'm representing that these two genes are linked. They're on the same chromosome, and I'm showing the allele combinations here. So this parent is big A, big B, and big A, big B. They are on the same chromosome. They are linked, okay? So we have a parental type. We always, in this parent, have a big A with a big B. In the other parent, we have a different combination. We have little a with little b and little a with little b, okay? So it's the same as a normal dihybrid cross, but what's really important to note is that we have big A with big B and little a with little b. Let's observe what happens in recombination to create non-parental types, which would be big A with little b or little a with big B. That's going to require recombination. So when recombination occurs, parts of the chromosome physically cross over and recombine. Now, this happens in the middle here, so you can see that we still have one big A, one big B gamete that's produced. We have a recombinant type, a big A with a little b, that's in there. We have a little a with a big B, another recombinant type, and then we have another parental type, the little a, little b. So what does this mean in terms of what we actually observe in the organisms? Well, again, let's back up and say these are not linked, right? If they're not linked, we should see equal distribution. We should see two parental type and two recombinant type, right? Because independent assortment says that that is equally likely to occur. But let's look at what happens when they are linked. All right, now we have the two genes on the same chromosome. If these genes are so close together that they do not recombine, no crossing over ever occurred, we would only ever see the parental types, right? This parental type was a big G, big L. We would only ever see that. And this parental type was a little g, little l. We would only ever see that in this test cross. We would only ever see parental types. What if recombination happened 100% of the time? Well, you might be saying, well, then we'd only see recombinant types. But that's not actually what occurs. Max recombination is 50% of the offspring being recombinant type. So you'll see 50% parental type and 50% recombinant type. Why is that? I just said it happens 100% of the time. Well, that's because of the way the chromosomes line up. The two inner ones are allowed to recombine, but the two outer chromatids here, they do not cross over. So the max recombination we can see is 50% offspring being non-parental. All right, so that would be they recombine every time. And in reality, you would not be able to distinguish this from unlinked, from on separate chromosomes. But what Morgan found was that sometimes genes have different strengths of linkage. So what he would actually find is that a certain amount would be parental types, but less than 50%, but more than 0 would be recombinant types. Okay, why is that? Well, the closer or farther apart that two genes are on the chromosome, the more, if they're farther, or the less likely, if they're closer, they are to recombine. And Morgan utilized that. He realized that if he looked at many traits in a cross, he could actually start to create genetic maps of the genes on the chromosomes. Again, this is just by using math, and him and his student Ch13 produced one of the first genetic maps in 1913. They did it purely by examining recombination frequencies. So here's Sturtevant with his flies. Of course, this is the early 1900s, so he is smoking a pipe in the lab, as many people did up until basically the late 80s, right? Here, though, this is not a real distance. It's not like this is 100 bases apart or 1,000 bases or 10,000 bases. This is in what we call centimorgans, which is a recombination value. How frequently do these things recombine? So here it's 13 centimorgans. So 13% of the time, these things will recombine. That linkage, that percentage there, allowed him to create these maps. How did he do that? That's a 300 level genetics thing as well. You have to examine 3 traits together and do some logic with it, but it's not that difficult, really. What I want you to take here is that the closer genes are, the tighter they're linked. That means they'll have a lower recombination frequency. If they recombine 1% of the time, we would call that 1 centimorgan. The possible range is from 0 to 50%, right? 50% means every time there is recombination, and that's the max distance we can get. Now, this is flies, right? And at the same time, people like Barbara McClintock here were working in other systems like corn. Barbara McClintock, very, very profoundly important individual in chromosomal theory. She studied very strange things that happen in corn chromosomes. She also proposed the concept of what we call jumping genes or transposons. And as many early scientists, and particularly female scientists, her ideas were not taken seriously until many, many years later when they were shown to be accurate. So jumping genes is an interesting topic we'll talk a little bit about later in the course, but she was able to link corn phenotypes with chromosomes as well. So we have good evidence from flies, we have good evidence from plants. Awesome. The kind of final nail in the coffin was being able to show linkage and chromosomes through a microscope. So in 1932, we have Kurt Stern, who he is able to observe that there's this unusual chromosome that had part of it broken off, and part of the X chromosome fused onto the Y chromosome. That trait that he was observing moved with it from the X to the Y, and he was able to show that the X now could recombine with that little bit of the Y chromosome because it had the same genetic material. So he was able to show that this is actually occurring in the microscope, and that was a really, really key point here. So if you want to learn more about all of these things, I'm just giving you like the 10,000 foot view here. If you want to learn more about this, Micro 250 is a great class. We spend a lot more time talking about the actual experiments here and looking at the pictures and stuff like that. So I want to finish with Mendel got really freaking lucky, right? He didn't encounter linkage. That's crazy, right? He had 7 traits and he had 7 P chromosomes, and he got lucky that each of his traits was on an independent chromosome. You can calculate the odds. I think it's 1 in 7 times 1 in 7 times 1 in 7 over and over again for 7 times, right? That's a huge value there. So he got really lucky that occurred. So Mendel did not live to see the basis for this discovered. Unfortunately, he died before this, but Mendel's work was rediscovered and contextualized in the view of chromosomes being the genetic material there. So chromosomes are important, but what part of chromosomes is important? Because what is the genetic material is the next question we're going to need to address, and we'll do that in a future chapter. Because chromosomes are made up of chromatin. And what did we say chromatin was? Chromatin is a mixture of protein and nucleic acid. That was a big sticking point there. Is it the protein? Is it the nucleic acid? Which part is actually the genetic material? That was a very big question. All right, so before we finish, let's do a review question here. You do a dihybrid cross between these parents. We have green fruit with a wrinkled skin in one of your tomatoes, and the other parent has orange fruit with smooth skin in the tomato. Okay, what would the non-parental combinations in the F2 offspring be? Go ahead and pause the video, think about that. All right, so non-parental combinations. Well, these are the parents, so we need the non-parental combinations. So in this case, it would be green fruit with smooth skin in the tomato and wrinkled skin with orange fruit, because that's the non-parental combination. The parental types are green fruit wrinkled and orange fruit smooth. Okay. Hopefully you are grasping that. If not, think about parent and non-parent there. Okay, so chromosomal theory of inheritance. Chromosomes are the genetic material responsible for Mendelian inheritance. There was a lot that went into this. Again, we just went over the surface of it. Evidence to support this: homologous chromosomes migrate independently in meiosis. We have them sorting. Each pair is random. Each parent has gametes with half the chromosome number. That makes sense. Sperm and egg have the same number of chromosomes. That all fits with the Punnett square and things like that. Gametes combine, and offspring have the same number of chromosomes as their parents. The cycle fits with Mendel's observations. Sex-linked traits in Drosophila were some really key evidence to support this, that we could actually observe those different chromosome types and traits moving with them. That was important. Some traits move together more often than not, and we call this linkage because they're on the same chromosome. The closer the genes that control the traits are on a chromosome, the tighter the linkage, the less recombination that occurs. And then remember, parental type, that's the combination of traits that's found in the parents. And then the non-parental type is the recombinant type, where we have the phenotypes swapped from what the parents had. All right, that's it for 13.1.